Module 2 with regard to the respiration in plants, the 14.4, the 11th Ncert. The heading it is aerobic respiration. In aerobic respiration, there is a complete oxidative breakdown of the pyruvic acid into carbon dioxide and water with the release of more amount of energy in aerobic respiration. In very first point, removal of all hydrogens from the pyruvic acid, leaving 3 molecules of CO2. It occurs in mitochondrial matrix. Second point, passing on of electron, removed as the part of hydrogen transfer to oxygen with simultaneous synthesis of ATP. What it is? Oxidative phosphorylation, where it occurs inner mitochondrial membrane. While passing on of electron, the proton transferred from the matrix to intramembrane space. Later, the proton from intramembrane space to matrix through F0 F1 complex results in the formation of ATP. So, passing on of electron, removed as the part of hydrogen, transferred to oxygen with simultaneous synthesis of ATP called oxidative phosphorylation, where it occurs inner mitochondrial membrane. Next, the pyruvic acid formed in glycolysis in cytoplasm that enter into the mitochondrial matrix through the proteins present in mitochondrial membrane. And the pyruvic acid undergoes oxidative decarboxylation, as it is undergoing oxidation, decarboxylation as well as condensation. And the enzyme complex involved, it is pyruvic dehydrogenase enzyme complex. In this cofactor, it is the magnesium, it is the cofactor. As from each pyruvic acid, 2 redox equivalents are removed and added to NAD. NAD got reduced to NADS2. So from 2 pyruvic acid molecule, 4 redox equivalents are removed, 2 NADS2 are formed. From 2 pyruvic acid molecule, 2 carbon dioxide molecules which are released, so decarboxylation, and it is condensed with coenzyme A. Pyruvic acid condensed with coenzyme A. So undergoing oxidation, decarboxylation, condensation with coenzyme A in presence of the enzyme pyruvic dehydrogenase and forms acetyl coenzyme A. That is the connecting link between glycolysis and the Krebs cycle. So what is the connecting enzyme between glycolysis and Krebs cycle? Pyruvic dehydrogenase enzyme complex. What is the connecting link? Acetyl coenzyme A. What is the connecting reaction between glycolysis and Krebs cycle? Oxidative decarboxylation. Which coenzyme forms connecting link? NAD as well as coenzyme A, which forms the connecting link. That connecting link is acetyl coenzyme A. This completes the link reaction. Now, acetyl coenzyme A entering into cyclic pathway, that is tricarboxylic acid cycle. What do you call it? Is Krebs cycle. Next, in tricarboxylic acid cycle, the Krebs cycle, acetyl group of acetyl coenzyme A condensed with the first member of the Krebs cycle, that is oxaloacetic acid, in presence of citric synthase by utilizing the water and form citric acid. Citric acid after dehydration and hydration isomerized into isocitric acid. The isocitric acid after oxidation and successive decarboxylation, it forms alpha ketoglutaric acid and succinyl coenzyme A. So successive decarboxylation product is succinyl coenzyme A. And during the formation of alpha ketoglutaric acid to succinyl coenzyme A, whatever the steps involved in the conversion of pyruvic acid to acetyl coenzyme A, same in this reaction. The alpha ketoglutaric acid undergoing oxidation, decarboxylation, as well as what it is condensation in presence of alpha ketoglutaric dehydrogenase and form succinyl coenzyme A. Next, the succinyl coenzyme A cleaved into succinic acid. During cleavage reaction, coenzyme A is regenerated out. In case of the very first reaction of the Krebs cycle, acetyl group of the acetyl coenzyme reacts with OA and forms citric acid. What is regenerated out? Coenzyme A. In this also, succinyl coenzyme cleaved into coenzyme A and succinic acid. During this cleavage process, the energy released couples GDP and IP to form GTP, guanosine triphosphate. And terminal phosphate of the GTP transferred to ADP, and ADP becomes ATP in a coupling reaction. By cleavage reaction, GTP is formed. By coupling reaction, ATP is formed. And the succinic acid undergoing oxidation to become what? Fumaric acid. The enzyme is succinic dehydrogenase and the coenzyme is FAD. The enzyme of the Krebs cycle not located in mitochondrial matrix but present as an integral membrane protein is succinic dehydrogenase. So succinic acid oxidized to become what it is? The fumaric acid with generation of FADH2. And fumaric acid, and it becomes the malic acid. Malic acid undergoes oxidation to become oxaloacetic acid. So regeneration of oxaloacetic acid completes the TCA, that is Krebs cycle. So pyruvic acid to acetyl coenzyme A, for 2 pyruvic acid 2 NADH2. In this cyclic manner, how many NADH2? 1, 2, 3 NADH2, 1 FADH2, 1 ATP. So for 1 pyruvic acid, how many NADH2 in mitochondria? 4 NADH2, 1 FADH2, 1 ATP, 3 CO2. In between pyruvic acid and acetyl coenzyme, 3 carbon, 2 carbon, 1 CO2. In case of the Krebs cycle, 2 CO2. So for 1 pyruvic acid, how many CO2 released in mitochondria? 3. For 1 pyruvic acid in Krebs cycle, how many CO2 released? 2. For 1 pyruvic acid, how many NADH2 formed in mitochondria? 1 in between glycogen and Krebs cycle, 3 in case of the Krebs cycle. So total it is the 4 NADH2. 1 FADH2, 1 it is ATP. So for each turn of the Krebs cycle or for 1 acetyl coenzyme A, 3 NADH2, 1 FADH2, 1 ATP, 2 CO2 are formed for 1 turn of Krebs cycle. For 2 turns of the Krebs cycle, 6 NADH2, 2 FADH2, 2 ATP, 4 CO2. So for 2 turns of the cycle, 4 CO2, 6 NADH2, 2 FADH2, 2 ATP. Suppose for 1 glucose in TCA, how many CO2? 2 turns. So it is the 4 CO2. For 1 glucose in TCA, how many NADH2? As for 1 cycle, 3 NADH2. Another cycle it is 3. Total it is 6 NADH2. So total we come across how many number of NADH2 in glycolysis? 2 NADH2. In between glycolysis and Krebs cycle, 2 NADH2, 4. In 2 turns of the Krebs cycle, 6 NADH2. Total 10 NADH2 and 2 FADH2 in case of Krebs cycle. So overall for 1 pyruvic acid in mitochondria, how many NADH2? 4 NADH2. How many CO2? 3 CO2. How many ATP? 1 ATP. For 1 pyruvic acid in Krebs cycle, 3 NADH2, 1 FADH2, 1 ATP, 2 CO2. So in Krebs cycle, there is neither involvement of oxygen nor large number of ATP formed. Why we are discussing about the Krebs cycle? No involvement of ATP. There is no production of, there is no involvement of oxygen. There is no production of ATP. Even then we are discussing the Krebs cycle. Why? There are large number of NADH2, FADH2 formed in mitochondria. That is in case of the Krebs cycle, which contain energy. The energy has to be released to synthesize ATP in a system, electron transport system. So whatever the reduced coenzymes in glycolysis and Krebs cycle undergoes oxidation in electron transport system and going to yield ATP in a process called oxidative phosphorylation. It is in contrast to the chloroplast where it is photophosphorylation, light-driven process. In this, oxidation reduction of the coenzyme drives the process of the ATP formation. What do you call? Oxidative phosphorylation. To recollect this information. So, how many oxidations so far it is? 6 oxidation. 1 in glycolysis, 1 in between glycolysis and Krebs cycle, and 4 in case of Krebs cycle. Next, how many total coenzymes are involved? 12 coenzymes. The 12 reduced coenzymes are found. In 12 reduced coenzymes, 10 are NADH2, 2 are FADH2. In 10 NADH2, in glycolysis, 2 NADH2, and in case of mitochondria, 8 NADH2. In 8 NADH2, 2 NADH2 in link reaction, 6 NADH2 in Krebs cycle. A total of 10 NADH2 and 2 FADH2. All the reduced coenzyme undergoes oxidation to release the energy. This energy couple ADP and IP to make ATP, that is electron transport system. Next, in electron transport system, the electron carriers, complex 1, complex 2, complex 3, complex 4, which are all electron carriers, which couple with the fifth complex, that is complex 5, that together constitute electron transport system. In electron transport chain, 4 complexes are involved. It combined with fifth complex and constitute electron transport system. Number of mobile carriers which are present in case of this mitochondrial membrane, that is the 2 mobile carrier. One it is ubiquinone within the membrane. One it is intermembrane space site, that is cytochrome C. In case of electron transport system, complex 1, what do you call? NADH dehydrogenase, otherwise NADH ubiquinone reductase. This complex 1 is involved in the transport of electron as well as proton. In this prosthetic group is FMN, flavin mononucleotide, and iron sulfur centers are present in complex 1. Next, complex 2. The complex 2 is succinic dehydrogenase complex. In this, FAD, flavin adenine dinucleotide, is the prosthetic group. So FAD is not only the coenzyme for succinic dehydrogenase, also form a prosthetic group in complex 2, as it is having iron sulfur center, and it is meant for only electron transport. Next, complex 3, that is called cytochrome C reductase, that is having cytochrome B, cytochrome C1 and iron sulfur centers. It is a complex 3. Complex 4. This complex 4 is considered as cytochrome C oxidase because it accepts electron from cytochrome C. In this, 2 copper centers are present, copper A, copper B, cytochrome A, cytochrome A3. In case of complex 3, cytochrome B, C1, in this cytochrome A, A3, and mobile carrier on intermembrane space side, the small mobile carrier only meant for electron transport is cytochrome C. In this, how many cytochromes are involved? A total of it is the 5. B, C1, A, A3 and cytochrome C. In this mobile carrier is cytochrome C. So how many total mobile carriers are present? UQ within the membrane and cytochrome C on intermembrane space side. Which mobile carrier is involved in electron and proton transport? Ubiquinone. Which is meant for only electron transport? Cytochrome C. The ubiquinone, it operates between 1 and 3 and 2 and 3. 1 and 3, 2 and 3. And cytochrome C operating between 3 and 4. So 2 mobile carries UQ and cytochrome C. So complex 1, complex 2, complex 3, complex 4 coupled with complex 5. Whatever the NADH2, FADH2 formed in respiratory process which undergoes oxidation, which is considered as the gateway for electron transport, it is the complex 1, NADH dehydrogenase. And number of ATP depends upon the electron carrier involved. If gateway for electron transport NADH, it is involved. As NADH2 undergoes oxidation to become NAD, electrons are transported to iron sulfur centers of, that is complex 1, and 4 protons are removed from the matrix translocated to intramembrane space. So, number of proton translocated from matrix to intramembrane space, it is the 4. And electrons transferred to the UQ, ubiquinone. The ubiquinone accept redox equivalents and got reduced to ubiquinol. And this ubiquinol reoxidized to ubiquinone by transporting the proton through complex 3 to intramembrane space. And during this, electrons are transferred to complex 3, iron sulfur protein. Then it is C1, then it is C. So complex 2 concerned with, complex 3 concerned with electron as well as what it is proton. So ubiquinol reoxidized to ubiquinone by transferring proton through complex 3 and transferring electron to iron sulfur protein. Next, from complex 3 electron transferred to cytochrome C, that is operating between 3 and it is the 4. And from cytochrome C transferred to copper A, cytochrome A3, copper B, and ultimately to oxygen. During this, 2 protons are translocated from matrix to intramembrane space. So for each oxidation of one NADH2, 4 plus 4 plus 2, 10 protons are translocated from matrix to intramembrane space. How many electrons are transported? 2 electrons are transported through electron transport chain. And how many water molecules are formed? 1 water molecule it is formed. So if you go for the pathway of NADH2 oxidized to become NAD, complex 1, UQ, complex 3, cytochrome C, complex 4, oxygen with formation of what it is? Water. So ultimate electron acceptor, hydrogen acceptor is oxygen in aerobic respiration. So for each oxidation of one NADH2, how many protons are translocated from matrix to intramembrane space? 10. How many electrons are transported? 2. How many water molecules are formed? 1 water molecule is formed. Of O2 it is involved. Next, with oxidation of one FADH2 is going to become what it is? FAD. FADH2 become FAD. And electrons are accepted by the complex 2. In this prosthetic group is flavin, that is adenine dinucleotide, FAD. In complex 1, FMN, in this FAD. From this electron transported to ubiquinone. It accepts redox equivalents, it got reduced to ubiquinol. And ubiquinol reoxidized to ubiquinone by transporting the proton through which complex? That is complex 3. Then to cytochrome C, then to complex 4, then to what it is? Oxygen with formation of 1 water molecule. The pathway of electron transport in case of FADH2, complex 2, ubiquinone, complex 3, cytochrome C, complex 4, and of O2 with formation of 1 water molecule. With oxidation of one FADH2, how many protons which are translocated from the matrix to intramembrane space? 4 plus 2, 6 proton, because it surpassed the complex 1. So there is a transportation of only 6 proton. So 1 water molecule it is formed. So with all the coenzymes, 10 NADH2 and 2 FADS2. In 10 NADS2, for each NADS2 10 proton, a total of what it is 100 protons translocated for oxidation of all NAD coenzymes for one glucose molecule 100. And 1 FADs to transport the 6 proton, 2 FADs to transport for it is another 6, so total of for it is the 12 proton. A total of 112 protons are translocated from matrix to intramembrane space with oxidation of all reduced coenzyme, that is 10 NADS2 to FADS2. So a total 12 coenzymes are involved. How many oxygen molecules are involved in this? For each NADS2 of O2, for each FADS2 1 of O2, there are 12 reduced coenzyme, 12 of O2, that means 6 O2 which are involved. How many metabolic water molecules are formed? The 12 metabolic water molecules are formed. So total of 112 proton present in case of what it is intramembrane space side. So much protons in case of intramembrane space and less number of proton in case of the matrix. So proton concentration gradient established from intramembrane space to matrix. And these proton tries to move from intramembrane space to matrix, but membranes are impermeable for transfer of proton. That less impermeability or no impermeability is the driving force for ATP formation. So which complex allows the proton from intramembrane space to matrix? Again, it is CF0 CF1 or F0 F1 complex. F0 is integral membrane protein. F1, it is peripheral membrane protein facing what it is, the matrix. As in case of chloroplast stroma, in this it is matrix. For every 2 proton down the gradient, it couples the ADP and IP to form ATP. In overall calculation, a dynamic calculation, for each NADS2, how many ATP are formed? 3. For oxidation of each FADS2, how many ATP are formed? It is the 2. So, for oxidation of one NADS2, 3 ATP. For oxidation of each FADS2, 2 ATP. Overall, the questions with regard to the electron transport system. How many complexes are involved in total electron transport system? 5 complexes. Only electron, 4 complex, which coupled with the 5th complex for formation of what it is ATP. Which complexes are concerned with electron and proton transport? Complex 1, complex 3, complex 4. Which complex concerned with only electron? That is complex 2. Which complex concerned with only proton? Complex 5. Which is the mobile carrier concerned with electron and proton? UQ. Which is the mobile carrier concerned with only electron? That is cytochrome C. How many number of cytochromes are involved? 5. In complex 3, B, C1. In complex 4, A, A3. And mobile carrier it is C. Total it is 5 type of the cytochromes which are involved. How many oxygen atoms are utilized? For 12 coenzymes, 12 of O2s, 12 oxygen atom. How many molecule? 6 O2. How many electrons are transported for oxidation of all reduced coenzymes? The 12 coenzymes, each a pair of electron, 24 electrons are transported through electron transport chain. And 12 of O2s are involved. 6 oxygen molecule, 12 metabolic water molecules are formed. See, next module, the calculating this number of ATP. So, how many number of ATP and what is the total number of ATP in case of the third module.